Welcome back. We're going to talk about more evolution in this section. This is Macroevolution Part 3, and today we're going to talk about Sympatric speciation and then a separate topic of punctuated equilibrium. So here we go. Hopefully you've been with me for the other discussions. We've already covered speciation in some detail with allopatric. We're going to contrast that a little bit with sympatric. But first, here's a quick review of three types of speciation. As always, you should pause and jot down the fascinating facts you see here on the screen and work at your own pace. I'm not going to read word for word every slide to you. I'll try to point out some particularly important points, but all of the text you'll see is important. On this slide, most important is allopatric speciation and sympatric speciation. Parapatric is probably not quite so important for AP biology tests, but it's certainly worth knowing because you're a biologist and you want to know how life works. But let's focus on sympatric speciation. So here we go. The key event, if you're with me last time for allopatric speciation, the key event always in making speciation possible is that a population has to be reproductively separated into two separate groups. By that I mean there has to be a way for separate pieces of the population to isolate their genes. Allopatric was easy. We did it geographically. We took a population and put them into two separate locations, and obviously they couldn't mix their genes once they were in separate locations. But in sympatric, sympatric means same homeland. Just like allopatric meant different fatherland, different homeland, sym means same homeland. So here we've got speciation somehow occurring within the same geographic location. So somehow we have to figure out how we can get a species, a population, separated into two separate groups, reproductively separated that is, without a physical separation. So they need to still be side by side, and yet for some reason the two groups can't mate with one another even though they're the same species. Once that happens, then it sets up the potential for evolution. As long as you can isolate gene pools, then those two gene pools have the potential to be treated differently by all the factors of evolution, in particular natural selection and genetic drift. And when those factors are at play differently in two different populations, it can possibly cause the populations to diverge from one another. And if they diverge enough that they can no longer mate with one another, then you have accomplished speciation. In some cases you're going to see in this discussion today it can happen in a single generation, but in most cases it might take a little longer than that, especially with animals. So let's try to look at ways in which we could have speciation occur within the same homeland. So here's one example. Just pause it here, jot this down. I'm not going to read this one to you, but it's a cool example. Okay, got it? Let's move on. Now I want to talk about one of my favorite stories of sympatric speciation, which involves the brightly colored cichlid fish that live in Africa's Lake Victoria. It is a really cool story because over the past few 100,000 years, from a single founding population of cichlid fish in this lake, there are now over 500 different species of brightly colored fish within the same lake. So sympatric speciation occurred. They were not physically separated, all in the same lake, and at least initially would be able to share genes across the whole population because the way fish reproduce in the water. But we now have over 500 species, and many of them certainly seem to have evolved through a process of sympatric speciation. So how could that happen? Believe it or not, it has a lot to do with physics, the physics of light, and with sexual selection. So the next several slides are going to explain some of this to you, the physics behind it, and then I'm going to get to the moral of the story, so to speak, after you've had a chance to read a little bit of the physics. So I'm going to ask you to pause on each of these slides, read it, maybe take a note or two. You're not likely to see any questions on the AP biology test about the physics of light, but it's a really cool story. It's one you might be able to use to illustrate a free response question to describe sympatric speciation if that shows up on the test. But again, the physics of light, probably not going to be on the test. I'll get to the moral of the story at the end, but I think you'll find this fascinating, how physics of light has interacted with biological organisms to lead to speciation within lakes. In particular, we're talking about Lake Victoria, although it could happen in any lake. But the cool story we're telling today are these cichlid fish in Lake Victoria in Africa. Okay, so take a minute and jot that one down. What I want you to notice here, in particular, we're talking about a situation with cloudy water. And just a little side note, in many cases, the cloudiness of the water is due to human interactions. So the activity of humans leads to the lake being cloudy, and that in turn could cause the physical effects we're going to talk about in this next series of slides. So it's always good to be aware of when human interactions are affecting living systems. Okay, hopefully you got this one. This middle bullet's the most important one on this page. This one we've already talked about. Fascinating. 500 species in a single lake in just a few 100,000 years. Got that one? Remember, slightly cloudy in this lake, probably due to human activity. Okay, key points on this slide. Fish that are sensitive to blue light are at an advantage in shallow water. Fish tuned to red light have an advantage in deep water. Okay, a little hint, that also means fish that have blue coloring will show up better in shallow water if it's cloudy. Fish that have red coloration will show up better in deep water. That turns out to be really important to this story. Okay, did you pause and get this one? Key point here, we're going to talk about sexual selection. You know what that is, we've talked about it before. In particular here, we are talking about intersexual selection because we're going to talk about mate choice. Okay, did you get that one? These are real pictures of these fish. That's really their coloring. I'm going to refer you to a cool YouTube video at the end where you can take a quick look at some of these fish in real life. But those are actual pictures of them. All right, pause and get this one down. Got it? Now here's my question for you. It says, sexual selection acting in this way. What are they talking about? What are we talking about on that slide? In what way? Can you explain it? If you're watching this with someone else, can you explain it to one another? If you're watching it on your own, can you explain it to someone else in your family? Can you explain what we're talking about here? What's the method of sexual selection that might be leading to speciation? Got it? So the moral of the story here is that blue males will find mates in shallow water, but if a blue male happens to migrate down into deeper water, he's not going to have any luck at all getting a mate. And the reverse is true for red males. Red males are much more successful in finding a mate and convincing a female to mate with them in deep water. These female fish choose the showiest, brightest, most brightly colored mate that they can find. So in deep water, the females choose red fish. In shallow water, the females will choose blue fish. And we know that the coloration is really what's happening here because in a really interesting experiment, some scientists took these fish of varying colors. They had females that they knew would normally choose red mates, females that would normally choose blue mates, put them in different aquaria with different colored fish, and it worked every time. The females that liked red males would only mate with red males, even blue males were around. And then they used a filter to affect the type of light that the fish were getting in each aquarium such that there was no coloration present. And if they filtered out the color so that the males all looked the same, then the females had no mate preference. So they would mate with either male. So that it is shown without a doubt it is the color of the males that was the key piece of the story. So the other key piece of our story for this presentation is that these fish live right next door to one another. So they are literally living in the same lake. They can swim past one another. They can go up and down in the water column. So it truly is sympatric, same homeland. They're living together. And yet the mate choices tend to force the populations apart over time. They diverge more and more and have at least the potential to produce different species, and that seems to be what has occurred here. So bottom line, mate choice and coloration can lead to sympatric speciation. So here's a little diagram of that. Great slide here. Probably the most important one up to now. And just one more time to summarize what the bottom line is here in this presentation on the sympatric speciation with the cichlid fish. And this really is a cool video clip. So if you're at home and you can get to YouTube, try typing that one in or clicking on it on your computer if it'll work for you, and watch that video clip. It's about a 12-minute discussion from a leading scientist who's researched this phenomena, and he describes not only the cichlids in Lake Victoria, but other examples where this has played a role. So that was sympatric speciation with animals involving mate choice. There are other mechanisms for sympatric speciation, and this one is a phenomenon called polyploidy. I'm sure you remember the words haploid and diploid. Haploid means a single set of chromosomes like we have in our gametes, like most animals have in their gametes. Diploid refers to two sets of chromosomes, which is what you have in all of your body except sex cells. So when a sperm and an egg join, they form a diploid cell, two sets of chromosomes. That's the normal state in animals, is diploid, except for their gametes. But in rare cases, an event called non-disjunction occurs. Do you remember what that is? If not, you should look it up. Non-disjunction. And in plants, non-disjunction is not nearly as detrimental as it is in animals. And what has been shown to occur in plants is that on occasion they will have non-disjunction of entire sets of chromosomes. And as a result, they get a gamete that is diploid instead of haploid. And since plants can often self-fertilize, imagine a plant that has non-disjunction of its gametes, then mates with itself, which is very common among plants. What's the offspring going to be? The zygote is going to be tetraploid, 4 sets of chromosomes, 4N. And in that case, you have an entirely new species in a single generation. And this is only successful in plants. You're not going to find animals that can be successful with 4 sets of chromosomes. Think about human genetic disorders like Down syndrome. Down syndrome is caused by a single extra chromosome, just one extra chromosome, and it's a small one at that, chromosome number 21. Imagine the detrimental effects of an entire extra set of chromosomes. It just doesn't produce a viable offspring. Doesn't mean it never happens in humans in the course of producing sex cells, but a zygote that is tetraploid or triploid in humans is not going to be a viable zygote. But in plants, it seems to work. And in fact, I guarantee you have seen and probably eaten plants that are polyploid, that are tetraploid. My favorite example are those giant strawberries. You know those really big strawberries, like they're 3 or 4 times a regular strawberry? Those guys are polyploid strawberries, multiple sets of chromosomes. Okay, so it happens fairly often in plants, not animals. There's another example, the primroses. Explanation of the process I just talked about. And if you prefer diagrams over words, here's a diagram of the exact same process we just discussed. So that was all, by the way, let's back up, sorry about that. That is specifically called autopolyploidy. Auto means self. So within a single plant, it's had this genetic event occur, non-disjunction of an entire set of chromosomes, mated with itself, and accidentally produced a new species in a single generation. The offspring cannot mate with other members of the population because their gametes will no longer match up. They'll have diploid gametes instead of haploid gametes. So that's self-polyploidy. That's by far the most common. But occasionally we have polyploidy that occurs when hybrids are formed, when two different plants mate and at the same time have a genetic abnormality occurring, a meiotic error like we see here. And this is allopolyploidy. Allo means different, like in allopatric. So different meaning two different species mated. And so this is the same result in the end. We end up with offspring that have a different number of chromosomes from the parents, and as a result, this offspring, when it produces sex cells after it matures, it will not be able to mate with other members of either population. So we've produced a new species, again, in a single generation, and in the same physical location. So it's sympatric speciation because they're living side by side with their parents and the rest of the population. So again, same geographic location, and yet they are genetically separate from the rest. They can't mate with them. So we have a population whose genes will not mix with the other members of the population. So we have the potential for speciation. In fact, this is speciation in every case because it's a brand new species in that one generation. I wouldn't worry so much about the details on a slide like this. The thing to remember, the moral of the story, so to speak, on this is that plants can have an extra set of chromosomes. It can have what would be many more chromosomes than what we'll call the normal state. That's called polyploidy. Results in sympatric speciation in a single generation in plants. So in your notes and in your mind, pair together polyploidy and plants with sympatric speciation. Another example of how sympatric speciation might happen with an abnormal chromosome number. This happens in the laboratory. It certainly occurred. Humans have been able to do it. Not likely to be an important part of the AP biotest, but you could discuss human-induced speciation, so human-caused evolution. And here's an interesting story. Chromosomal rearrangements in animals that produced viable offspring. Now this is not a total non-disjunction, but a fusing of chromosomes, so a change in chromosome structure where there was still a functioning zygote, but produced enough differences in their chromosomal arrangements that they couldn't mate with the parent population. So we had a new species in a single generation due to, let's roll back to the title of this one, due to chromosomal rearrangements. Okay, so there are rare cases where we see it happening in animals, but it's not polyploidy. It's fusing or a breaking of chromosomes. So something has happened to the chromosomes to change their number, but it hasn't changed the information available. It's just changed the way the DNA is packaged. And because the cells go through meiosis to produce sex cells, if these two grasshoppers in the picture have a different number of chromosomes, then there's no way they could successfully mate, even if they have exactly the same genes. If those genes are packaged differently in their chromosomes, you've got new species. So that's the way we have genetic abnormalities resulting in new species of animals. It seems to be fairly rare, but it does happen. But again, it's not the same thing as polyploidy. Okay, let's switch gears altogether. So now we're talking about not sympatric speciation, but looking at patterns of speciation in the fossil record. And for this discussion, we're not worried about whether it was sympatric or allopatric. In most cases, it's going to be allopatric speciation. But here what we're talking about are two different models that evolutionary biologists discuss and debate that relate to the tempo of evolution, how fast evolution occurs. The first model is called gradualism. It's by far the model that's been discussed for the longest time. That's the main thing Charles Darwin talked about, and most biologists since the 1850s when Darwin published On the Origin of Species. When they talk about evolution, they're talking about natural selection resulting in the slow, steady accumulation of changes over time, and with those accumulation of changes eventually leading to differences that are significant enough from the parent population that you would have to give this species at that point in time a new name, be a new species. So here we're looking at a simple version of it using coloration in butterflies as a way for us to visually observe the changes that are happening over time. Naturally, in fossil record you wouldn't see these changes, but this is just a way to visually represent it. We're not necessarily talking about butterflies, such as the example here. So imagine we have an ancestral population that I'll circle, and let's just call it population A. And over time it diverges, so probably allopatrically diverges, meaning we're going to two separate geographical locations. And now in the square here and the square on the right, they're physically separated from one another. And through the normal mechanisms of evolution, natural selection, genetic drift, sexual selection, all those 5 mechanisms we talked about before, in these two separate locations, they gradually accumulate changes over time, and by the end of this chart, at the top of the page, we've got 2 different species arising from A. So we can say this is species B and species C. And they came about by the normal mechanism of natural selection, gradually accumulating variations over a period of probably millions of years. So again, you wouldn't necessarily be looking at butterflies, but looking at fossil organisms and noticing changes in their structure over multiple layers of rock, gradually accumulating changes until they were so different you simply had to call them different species. Okay, so that's gradualism. A different view of the rate of evolution was proposed in 1972 by 2 scientists. You should recognize their names and be able to recognize their names on a test, Stephen Jay Gould and Niles Eldredge. They proposed a mechanism that they called punctuated equilibrium. They were using this term to describe something they had observed in the fossil record. Their observations were that many times in the fossil record a species tended to stay the same for millions of years, and then in what seemed to be a geologically rapid period of time, in a single layer of rock, that ancestral population would give rise to a whole new species. Clearly from that ancestor population, you could tell that the previous population were the parents, so to speak, of this new species, but the new species appeared fully formed in a single layer of rock. And then once it was there in that new layer of rock, it was found in numerous layers of rock for many millions of years again. So in the picture here we have, let's call it original population A. And in this model, just like in the other model, occasionally you might have one species that doesn't really change enough to get a new name. So over millions of years we still have species A. But as we go through these layers of rock, let's say these are layers of rock, these lines I'm drawing. And imagine you're digging fossils, and in layer 1 you only find species A. In layer 2 you only find species A. But in layer 3, all of a sudden you not only find A at this depth, but you also find species B. Boom! Species B is there in layer 3. It wasn't there in layer 2. Okay, so this model is proposing 2 points. One, that a species tends to stay the same for a long period of time. That's the equilibrium part of the name. Okay, equilibrium, staying the same. But that every now and then something really special happens in the environment. We'll talk about what that might be in a minute. But something special, something unique happens and causes evolution to happen so fast that in the next layer of rock, species B can appear, a new species can appear. So this equilibrium was punctuated, like punching a hole in it, was punctuated by a rapid change, and now speciation has occurred. So the term punctuated equilibrium refers to 2 things. Equilibrium is staying the same for long periods, and they called that stasis, staying the same. Every now and then it is punctuated, boom, by a change that results in a new species. Now notice A could still be there. And so now the next layer of rock, we've still got A, we've still got B, and then in the very next layer of rock, boom, now we've got species C showing up. We still have A, still have B. And now, once B is established, notice on the right, it stays as B for millions of years. So that's the equilibrium part of the name again. Species C, boom, punctuated the equilibrium of A by suddenly showing up in the next layer of rock. And as a result, we now have a status quo, a stasis situation for that species. Let's get rid of that. Here we go. All right, so that's the idea of punctuated equilibrium. Staying the same for long periods of time, punctuated by geologically rapid speciation. Now, what I want to make clear is that punctuation is only rapid in a geologic sense. So when we say geologically rapid, we're talking about 100,000 years. So to you or to me, it would not seem rapid at all. So it's not literally overnight like the sympatric speciation with polyploidy we talked about. That's not at all what we're talking about here. It's a change that is still very slow in human terms, but to a geologist, it's the next layer of rock. Okay, so it could be 100,000 years for the speciation event, but to a geologist, it's a rapid event. Okay, so what are some things that could account for this? How do you suppose it might occur? Well, what's happening here is just a factor or a result of the fact that something special has occurred in the environment that would speed up the process of natural selection. So here's some things that might occur. Imagine that the population diverged and from the edges. So let me do a little drawing here. Let's say this is the range of our population where species A is existing. And then over here on the edge, right here, I'm going to make a little dark circle. The members of the population over here on the edge, they're at the very extremes of the range where this species can be successful. And for whatever reason, something breaks them off. Maybe there's a river that cuts through. Some sort of event separates this fringe population from the main group. Now, several things have happened when we've done this. Now we have a small population, so that sets up the potential for genetic drift, where random events can have a significant effect. It also means we have a small sample of the whole population, so they're probably not genetically the same as the rest of the group anyway, because it's a small group. And because they're on the fringes, they're able to survive at the very edges of the range, so they're probably a little bit different genetically from the rest. Now they're separated here, so we've got a geographic separation. So now we can have allopatric speciation occur, but it's occurring with a small subgroup. In a small group, natural selection happens much more quickly than in a large group. With a really large population, hundreds of thousands of individuals, natural selection is very, very slow. It takes many, many, many generations to accumulate enough change that you would even be able to notice it, because most of the individuals have the same basic genetic makeup because there's such a large population. So even in a really good mutation in one or two individuals within several 100,000, it's going to take a long time for them to have enough offspring that the population would look different. But in a small subgroup, natural selection can occur much faster. And as a result, you could lead to a new species much faster. So that's what we mean by some special event in the environment. Something has to separate a small subgroup and allow natural selection to have its impact much more faster. Okay, so we have this period of equilibrium, and then something special happens, boom, punctuation, which produces a new species, then right back to equilibrium again for a long period of time. It's important to note this last bullet. The fossil record shows both types. There's lots and lots of evidence of gradualism occurring in the fossil record. There's also evidence of punctuated equilibrium. There are debates among evolutionary biologists about this idea, and some would say that punctuated equilibrium is just something that appears to happen because we haven't found enough fossils. If we could find more fossils, we'd see all the gradual changes. But there's also a large group of evolutionary biologists who accept punctuated equilibrium as a real phenomenon, but it occurs in, but we see both occurring in the fossil record. So how are they really different from one another? I've said both involve natural selection, and in fact, when I first learned about this model of punctuated equilibrium many years ago. I worried about it, I'll be honest with you, because it seemed to be counter to anything I knew about evolution. And I really wanted to know how it worked. And by good fortune, I got to meet Stephen Jay Gould, one of the authors of this plan. We had dinner together, and it's a long story how it happened, but it was a cool thing for me as a biologist. And I got to sit down at the dinner and ask him about this idea, and I wanted to know, was his idea of punctuated equilibrium different from Darwin? Was it really arguing against what Darwin had said? Was it anti-Darwinian? And he laughed at me and said, no, it's not. But let me explain to you what's happening. So here's what he showed me. In the gradualism model, we've got our species A here that's going to diverge into B, and let's say this one stays as A. So what he said was the first thing that happens is the population has to split right at that fork, and if we could look at individuals in that population where the arrow is, or in this population where the arrow is, both of those would still call them A. But on the left, let's say it's starting to change a little bit because of natural selection and it's in a new location. So let's call it A primed. It would still be a subspecies of A, still looks a lot like A. You'd be hard pressed to give it a new name, but it's a little different. So we're going to call it A prime. Then we move up a little further. Let's call this one A double primed. On this side, it's going to be A all the way. On this side, pretty soon we've got another change, A triple primed, and so on. So gradually accumulating change. What if we looked at the one right below B? So where the B is represents where it's different enough that we agree we're going to give it a new name. It's a new species, clearly physically different enough from A. But what would it be like right below B? It'd be a lot like B, but not quite B yet. So let's call this one B prime. And the next one down, B double primed. Here is B triple primed. Hopefully you're getting the idea. At some point, we have to switch names from saying it's more A-like to more B-like. And we'd see along the way here this slow, steady, gradual accumulation of differences. And if we had a time scale on here to get from the bottom A to the top B, might be 10 million years. Okay, 10 million years. Now, on that same time scale, if we looked at punctuated equilibrium, here is A staying as A for a period of 10 million years. But in punctuated equilibrium, in one layer, all of a sudden, we have B showing up and staying as B for a long period of time. So what's the difference? The key is we don't see all of the A primes, A double primes, A triple primes, B triple prime, B double prime, B single prime, B. Where would all those changes be? In my drawing on the right, where do you think all those changes would have existed? Remember, it took 100,000 years. And over the period of 100,000 years, those changes would have been occurring. So I said to Stephen, well, what if we took the graph on the right, which I'll represent with a star, and instead of representing 10 million years from top to bottom, what if we drew it out of a scale of 120,000 years instead of 10 million years? What would this graph look like? What do you think he said? Have you figured it out? What did he tell me? If I represent 120,000 years instead of 10 million years, what do my lines look like? If you figured it out, he told me it looked just like this first one. It looks like gradualism if we could only see every change over 100,000 years. So we see this same pattern, and this branches out like that. If it's 100,000 years from here to here, we would still see A prime, A double prime, A triple primed, and so on. But because all of these changes were compressed into a period of 100,000 years, and because that's the length of time it took to make a layer of rock, we don't find very many of those fossils. We probably don't find any of the in-between fossils. We might find a few if we're really lucky. But if the forms of A and B existed for 10 million years, and you're looking at layers of fossil, what are you more likely to find, A and B, or A primed, A double primed, A triple primed? That's how he explained punctuated equilibrium to me. Because of stasis, you're much more likely to find these fossils, all of these Bs that are not changing for long periods of time, and you're much more likely to find all of these A's. All the other varieties over here on the left, they did exist. There was a period in time where there was an A primed, an A double primed, an A triple primed, but they all existed for a total of 100,000 years, all of them together. So we're not likely to find those fossils. We only find fossils, or we're more likely at least to find fossils of the established species. Okay, so the moral of the story for me was that the mechanism is exactly the same in punctuated equilibrium as it is in gradualism. The difference is the rate, how fast it occurs, because of some special event that separated the population into a small subgroup where evolution could occur rapidly. So he wasn't proposing any mechanism different from what Charles Darwin had proposed. He was just proposing special cases, like maybe moving to an island. And Darwin actually proposed something very similar. He didn't call it punctuated equilibrium, but he described island evolution as being a special case that would occur much more rapidly. Okay, so I hope that made sense to you. One more look at those pictures. And here's another way to represent it, two different diagrams. And again, the scale is all about time. So the difference between gradualism and punctuated equilibrium is all about rate, how fast it occurs. It's not about how it occurs. Not about mechanism, but how fast. All right, that's it for that presentation. I hope you found that useful. We'll talk to you next time. I think we have one more talk to go together with evolution. We're going to look at the origin of life next.